Hello all, welcome to UNext Officer channel. I am Dr. Adira and I will be teaching you important topics of biology which are repeatedly asked in central exams. If you haven't subscribed this channel, then please do subscribe this channel. So in today's episode, we are going to discuss about RNA, its structure and difference between DNA and RNA. So first, what is the full form of RNA? RNA is ribonucleic acid and there are mainly three types of RNA. They are mRNA, tRNA and rRNA. So first let's discuss about the mRNA. This mRNA acts as a messenger between DNA and protein production during the process of translation. Now about the tRNA. This tRNA, the RNA is folded into a T-like shape and hence the name. The function of the tRNA is to bring amino acids together to form protein during translation process. And this rRNA, this R stands for ribosomal RNA, and the function of the rRNA is it is usually a main component of the ribosome, and this ribosome function is to read mRNA to form proteins during translation. So these three are the different types of RNA. So let's now discuss about differences between DNA and RNA. So the first difference is in the structure. As you can see in this image, DNA is always double-stranded, whereas RNA is always single-stranded. So this is the main difference. Then they also differ in the size. DNA is usually larger than RNA and it has millions of nucleotides, whereas RNA has only hundreds of nucleotides. Now, next difference is in the sugar present in them. The DNA usually, as I have already said in the earlier video, in DNA the sugar is deoxyribose sugar, whereas in RNA the sugar is ribose sugar. So I'll explain about that point. Let's look into the structure. So this is the structure of DNA and it has phosphate group. So it has a ribose sugar, it has a phosphate group and a nitrogenous base, and in the second carbon atom, this is the second carbon atom. In DNA, the second carbon atom is attached to the hydrogen, whereas in RNA, similarly ribose sugar is attached to phosphate group and it has a nitrogenous base, whereas the second carbon atom is attached to OH group. So this is RNA and this is DNA. So that's the difference in the ribose sugar. Now coming to the next point, that is location. The DNA is usually present in the nucleus of the cell, whereas RNA it is usually found in cytosomes or in the ribosomes. And then they differs in the nitrogen bases. In DNA, the nitrogen bases are adenine, guanine, cytosine and thymine. Whereas in RNA, the ribose sugars are adenine, guanine, cytosine and uracil. So it is important to note that the RNA, in RNA, uracil replaces thymine. So that's the difference in nucleus bases. Now coming to the last point, that is expression of genetic information. In DNA, the genetic information contained in DNA will be carried on to mRNA, and this mRNA will result in protein synthesis. So the process in which DNA is converted to mRNA is known as transcription, and the process by which mRNA is converted into protein is known as translation. So hence the protein is synthesized in our body according to the genetic information in DNA. But in those species in which the RNA is act as a genetic material. That is, in certain viruses, RNA act as genetic material. So in them, how will the information transferred for protein synthesis? In them, RNA is first converted into DNA, and from this DNA they can be further carried on to the protein synthesis. So from DNA, mRNA will be formed, and from mRNA protein synthesis occurs. So here RNA is being converted into DNA first. This is the initial step, and this is known as reverse transcription. So in those species in which the RNA acts as genetic material, this genetic information is carried out through reverse transcription. So that's all about today's topic. Hope you have all have understood today about today's topic. If you have any doubt, then please do comment it in the comment box below. And let's meet in a next video with another topic. Thank you guys.